A couple of topics ago, we talked about Mendelian genetics and how Mendel saw these really simple ratios in his experiments. Again, he got these simple numbers because he was dealing with cases where there were two alleles, one was dominant to the other, and he was only working with the effects of one or two genes. As mentioned in our last topic, non-Mendelian genetics, this is not usually the case. Most traits that we can observe are caused by the effects of many, many genes working together. And today we're going to talk about how those genes are regulated. Why do cells need to regulate genes? Well, first of all, they need to conserve resources. In a cell, there might be a gene that's not needed at that point in time. Perhaps we're talking about a cell that can take in lactose, which is a sugar, and use that as a food source. But maybe there's no lactose in the environment. If that's the case, there's no point in that cell making the enzyme lactase, that's the enzyme that breaks down lactose, there's no point in making that protein if it's got nothing to do. It would be a waste. So that gene might be turned off. When cells develop in a multicellular organism, they change roles. They change identities as you develop as an embryo and then a fetus. Different genes are needed at different steps along the way. If we look at any of the cells in your body, let's say a skin cell and a heart cell and a liver cell, all of those cells have the exact same information. Every one of those cells has the same set of chromosomes with the same genes on them. But of course, those three types of cells do very different things. The heart cell doesn't need a lot of the information that's needed by the liver cell. So even though all the information is there in all of those cells, the genes that aren't needed are shut down. So in a heart cell, we're going to see a certain set of genes that are turned off that might be turned on in a liver cell. We'll have a certain set of genes that are turned on in the heart cell that maybe aren't turned on in the liver cell or the skin cell. So we need to regulate what genes are being used. Let's start by talking about gene regulation in prokaryotes. Remember that prokaryotes don't have a nucleus. So we're talking about bacteria and archaea here. There's no nucleus, and the cell itself is simpler because we don't have compartments like the Golgi body, etc. We also have very, very little junk DNA. So in our cells, about 96% of your DNA doesn't appear to code for anything. It may have other functions, we'll talk about that a little bit later, but it doesn't seem to code for messenger RNA that's used to make proteins. Prokaryotes, like bacteria, are very efficient. Generally, all their DNA does something. They also have their DNA in a great big circle. So unlike in eukaryotes where we have separate linear or straight chromosomes, they have most of the information on one great big circle, one circular chromosome, and then additional information found within plasmids, which are little circles of DNA. Transcription and translation also happen very, very quickly, and that means these cells can prepare for division very, very quickly as well, and that's why bacteria can divide and spread so quickly. Prokaryotes are very good at organizing their information. So let's say that we had a metabolic pathway and it's got 5 steps to it, and we require 5 enzymes, one for each of those steps. There's going to be 5 genes, one that codes for each of those enzymes, and in prokaryotes, they'd all be next to each other on the chromosome. Whereas in eukaryotes like our cells, those genes might be scattered all over the place. They might be on totally separate chromosomes. In bacteria, quite often, all of the genes that are needed for one pathway are right next to each other, and all of those genes are transcribed as one great big long messenger RNA. That's known as a polycistronic messenger RNA. That's going to find a ribosome, and the ribosome will bind, and the ribosome will bind to one of the starting locations on one of those genes and decode it into a messenger RNA. And that's what you're seeing here. So we've got ribosomes that are going along, almost like an assembly line. They're going along that great big messenger RNA, and they're spitting out separate polypeptides. So separate proteins that will then be folded and give rise to these separate enzymes that are all going to work together. Now, the reason this makes sense is that you can shut down that entire polycistronic messenger RNA. You can shut down the production of that when you don't need to undergo this particular metabolic pathway. And I'll show you what I mean in just a moment. Another way that prokaryotes are really efficient, and you saw this on the last slide as well, is that they can have multiple ribosomes bound to a messenger RNA at the same time. So what you're seeing on the left here, we've got ribosomes coming in on the far left. So we've got the large and small subunit coming together and clamping onto the messenger RNA. And then they travel along and they fall off the other end. But before they fall off, another one comes in behind them. So we have this assembly line of ribosomes that are moving along and spitting stuff out as quickly as possible. In eukaryotes, there is, of course, a nuclear envelope. There's a nucleus. The messenger RNA has to leave the nucleus. It has to go through a nuclear pore. It has to travel out into the cytoplasm, and it has to find a ribosome. And that takes a little while. But because there is no nucleus in prokaryotes, and because all of the enzymes and everything that's mixed within the cytosol is in direct contact with the DNA, transcription and translation can occur at the same time. So what you're seeing here is we have RNA polymerase. Remember, that's the enzyme that makes messenger RNA. It's traveling along a DNA molecule, and it's spitting out a messenger RNA. And at the same time, before RNA polymerase is even finished, the messenger RNA is being translated by ribosomes, by these polyribosomes, this assembly line of ribosomes. In that photograph, or micrograph, at the top, you're seeing what this looks like under an electron microscope. And what we've got here is we've got the DNA along here, continuing along there, and then we've got messenger RNA that's being produced by RNA polymerase, and we can see that here. And then those circles are ribosomes that are already attached to the messenger RNA, and they're translating it. They're turning it into protein. Because this can happen at the same time, it greatly speeds up the rate at which bacteria can spit out protein and get ready for things like cell division. What we have here is a simple biochemical pathway that's found in bacteria. And this is the pathway that's used to make tryptophan. Tryptophan is an amino acid. It's something that the bacteria cell definitely needs to make. But if a cell is making tryptophan, it will get to a point where it has enough and it doesn't have to make any more. So to be efficient, there needs to be a way to shut down this pathway. Let's start by looking at the left-hand side of the diagram. So we have our precursor at the top, and we have our end product at the bottom, tryptophan. There's 3 steps to this process, and they're each catalyzed by a different enzyme. And you can see the genes that code for those enzymes on the right. Now, what's interesting about this pathway is that tryptophan can actually shut down enzyme 1. So if tryptophan reaches a critical concentration, it will bind to any of the enzyme 1 that's floating around and shut it down. So if you remember, we talked about allosteric inhibition of enzymes. That's where something binds to the enzyme outside of the active site and changes the shape of the active site. And that's what tryptophan does. So a nice, neat, simple system where the end product shuts down the first step. But it's still not quite as efficient as it could be. So in that first example, the biochemical pathway was being shut down when it was no longer needed by this negative feedback inhibition. However, the genes that code for those enzymes are still going to remain active. They're still going to be used to make messenger RNA. The messenger RNA is still going to be translated. We're still going to make those 3 enzymes, and that seems like rather a waste. So another way that this can be regulated is at the gene level. So let's take a look at those genes. So we have TRPE, TRPD. They each code for one polypeptide, one protein. Those have to come together to make enzyme 1. Then we have the TRPC gene, which codes for the enzyme number 2. And then we have TRB and TRPA, and those code for enzyme 3. So this is another case where the 2 proteins have to come together to make that 3rd enzyme. What if we could shut down all 5 of those genes? That would save us a lot of time and effort, or it would save the cell a lot of time and effort, and a lot of waste. It would save a lot of waste because we don't have to make those messenger RNAs, we don't have to make those enzymes. We can just shut everything down right at the start. Bacteria are able to do this genetic regulation using something called operons. An operon is the basic unit of gene expression in bacteria. So basically, all of the required genes for a particular pathway are all grouped together. Just in front of that, we have something called a promoter. And the promoter is essentially an on-off switch. If it's turned on, RNA polymerase is able to bind, and it's going to move along those genes. So in this case, we have 3 genes. And it's going to spit out a messenger RNA that has the complementary sequence for all of those genes. And we can see our messenger RNA here. Now, each of those genes has its own start and stop codon. We talked about that before. So in the messenger RNA sequence, we have these green areas, which are starts. We have these red areas, which are stops. And what's going to happen next is a ribosome will bind at that start location, and it will move to the stop location. And that's going to happen individually for each of these gene sequences on the messenger RNA. So we're going to get 3 different proteins being spat out during translation. Let's go back and take a look at that tryptophan pathway, but this time we're just going to focus on how it's regulated genetically, not how it's regulated through negative feedback biochemical inhibition. So we have all of the needed genes, all 5 of those genes needed for the enzymes. We have them all grouped together, and they're under the control of a single promoter, a single on-off switch. So what will happen is RNA polymerase, that's this egg-shaped thing right here, will bind at the promoter, and then it's going to run along and it's going to transcribe that into one great big long messenger RNA. And once again, the messenger RNA is going to have 5 gene sequences in it, and each one of those is going to have a start codon and a stop codon. And that's going to be turned into 5 different proteins. We're going to have these 2 coming together to make one enzyme, and these 2 coming together to make one enzyme, and then we've got C on its own functions as an enzyme. So that's our promoter. That's where RNA polymerase is going to bind. But right next to that, we have something called the operator. So the promoter is made up of the binding site where the RNA polymerase is bound, and the operator that can bind to a regulatory protein. So let's take a look at what's over here now. So we've got a break in the gene here. Those 2 lines represent that these 2 things are not necessarily close to each other. Somewhere else on this big circular chromosome, we have another gene that's a regulatory gene, and it's known as TRPR, the R for regulatory. It codes for its own protein, and that's the protein we see down here. And this protein if tryptophan is present, will change shape and bind to the operator and block RNA polymerase. So let's just go over that again. We've got our operon, which is the promoter. The promoter consists of a binding site for RNA polymerase and this operator where the regulatory protein can bind. And then we have the 5 genes. So the promoter and the 5 genes make up the operon. The default setting for this is on. If there's no tryptophan in the cell, then this is on, and the cell is manufacturing the proteins, the enzymes it needs to undergo the tryptophan pathway. Meanwhile, elsewhere on that giant circular chromosome, we have a totally separate gene. It's not part of the operon. This is a regulatory gene. So the gene is known as TRPR, and it codes for a repressor protein. And this repressor protein starts out inactive. So what I mean by that is it's not repressing anything, it's not doing anything. But that changes if tryptophan is present. If tryptophan is present, it will bind to that repressor protein. And that repressor changes shape. It's now being activated. It can actively repress the operon. So it's going to travel to the operon and bind there, and no RNA is going to be made for any of these 5 genes. And the reason for that is that RNA polymerase is physically blocked. RNA polymerase cannot move to the right in this diagram because it's physically blocked by the binding of that repressor. So we have successfully shut down this whole pathway right at the genetic level. The tryptophan operon is referred to as a repressible operon. What I mean by that is it's typically turned on, but it can be repressed, turned off, if there's enough of the end product. So a repressible operon is involved in manufacturing something that the cell needs, and when there's enough, the operon is repressed. There are operons that work in the opposite way. They're inducible, and the classic example would be the lac or lactose operon. Here we're not talking about building something, we're talking about tearing something down. So lactose is a sugar, and cells can break it apart and get energy from it, but there's no point in making all the proteins needed to do that if there's no lactose. So let's imagine an E. coli bacterium in your gut, in your intestine. If you drink a glass of milk, all of a sudden lactose is present. And it would make sense for the E. coli to start preparing to break that down. The lac operon has 3 genes, and these are genes that are involved in breaking down lactose, and also there's a gene that's involved in creating channels within the membrane that can be used to bring lactose into the cell. But again, we don't need to make any of that stuff unless lactose is present. So if lactose is not present, again, we have this lac I, it's called, for inducible. We have this regulatory protein that's going to be manufactured. There's a separate gene that codes for that, and that can bind directly to the operator of the promoter and block any sort of transcription. However, if lactose is present, it stops that repressor from binding. So in this case, when lactose is present, the lac operon is turned off. So normally if there's no lactose, that repressor protein is bound to the promoter. But if there is lactose, lactose binds to that regulatory protein that's already bound, and it causes it to fall off, and it allows RNA polymerase to move down and transcribe all of those genes. So again, we have 3 proteins that are going to result. We have this beta-galactosidase. Don't worry about the names of these enzymes, but that's an enzyme that breaks down lactose. And then we have permease, which makes the cell more permeable to lactose, and something called transacetylase, which also assists with getting lactose into the cell. Moving on to eukaryotes. Eukaryotes are messy. They're sloppy. They're not nearly as efficient and organized as prokaryotes are. First of all, instead of one great big circle of DNA, we have lots of linear or straight pieces of DNA. But more bewildering, only 2% of our DNA codes for proteins. So what's the rest of the DNA? Well, some of it is promoters. Those are on-off switches for genes. Some of it consists of enhancers. Those are sequences of DNA that enhance the rate at which a gene is transcribed. We also have introns, which are non-coding bits of DNA that are actually found within genes. So even the genes have these bits of non-coding DNA inside them. We have sequences that code for transfer RNA, ribosomal RNA. There's other types of RNA as well, small nuclear RNA, and etc. We're not going to talk about those in this course, but we will talk about telomeres. Telomeres are sequences of DNA that are found at the ends of the chromosomes. And they're actually there as kind of a stopgap to help prevent the erosion of chromosomes when chromosomes are replicated. So every time your chromosomes are replicated, you lose a bit of DNA. And we'll talk about why that is when we talk about DNA replication in our next topic. But telomeres are really fascinating. They kind of hold the secrets to longevity, as we'll see. And then the rest of the DNA, almost 96% by some accounts, is so-called junk DNA. Now, it might not be completely junk, but we don't know precisely what the function is. Because eukaryotes are kind of messy and sloppy when it comes to organizing and decoding their DNA, regulation of DNA activity is more complicated in eukaryotes. Remember that we have a nucleus. We have 3 broad steps where DNA expression can be regulated within a eukaryotic cell. We have before transcription even occurs. We have regulation at the transcriptional level, which is the most important one. We have also regulation of how the messenger RNA is processed. Then once the messenger RNA gets to the cytoplasm, there are some controls that act on the actions of the ribosome. So those are translational controls. And then finally, proteins are usually modified after they're made by a ribosome. So they have to be folded, they have to have sugars added to them and so on. And all that stuff will determine what that protein ends up doing. When a gene is used to make a protein, and that protein does something, it changes the cell somehow, we say that the gene is expressed. Let's start at the beginning. How do we control gene expression in eukaryotes? We can start before transcription even occurs. So remember, chromatin is the material that makes up chromosomes. It's a mixture of about half protein, half DNA, and the DNA is attached to those proteins. The proteins condense the DNA prior to the division of the cell. We've talked about that occurring. If we have very tightly packaged DNA, it's referred to as heterochromatin. And this is DNA as we would see it, let's say, during late prophase or metaphase of cell division. When the DNA is that tightly compacted, RNA polymerase and transcription factors, those are proteins that help this whole process, can't get to the DNA. So the RNA polymerase can't get to the promoter. It can't physically get to the gene to transcribe it. Now, what happens in some cells is they can permanently deactivate the genes that are present that they don't need to use. So I mentioned before, heart cell doesn't need to use some of the genes that a liver cell does. So those genes can be turned off. And what the cell does is it sticks methyl groups to those genes, and that prevents RNA polymerase from binding, and it also activates the histones and other proteins that are needed to condense that DNA very, very tightly. This is something known as epigenetics, which I won't get into right now, but it's the idea that you can turn off certain genes by compacting them, and that's actually inherited. When the gene is when the cell is replicated, those turned off genes will remain turned off in the daughter cells as well. There's some evidence to suggest that doing some nasty things like smoking, for instance, might permanently turn off some genes, and those genes might be turned off in daughter cells that result from that cell. So that's kind of interesting. If we have time, we'll come back to that later in the course. In any case, we have two types of chromatin. We have U chromatin. U means true. This is the chromatin that can be read by RNA polymerase. It's loosely attached to histones, and the genes within U chromatin can be used by the cell. Then we have heterochromatin, where the DNA is very densely compacted, and this is in between cell divisions, and the DNA is so compacted that RNA polymerase can't get to it, and those genes are unreadable. So in this cell here, we have a few interesting things going on. This is an electron micrograph, so very, very high magnification. You can see some of the organelles we've talked about in the past, the mitochondria, the rough endoplasmic reticulum, the Golgi body or Golgi apparatus or Golgi complex. And then also in the nucleus, which is very large in this cell, we've got two different types of chromatin. We've got U chromatin, which is pretty open, and it contains cells that can be read. And then we have heterochromatin, which is very highly compacted, and those genes can't be read. And this is going to be passed down to the cell that results as well, because the cells have these methyl groups attached to the DNA where it's highly condensed, and those methyl groups are copied as well. Transcriptional control is the big one when it comes to regulating gene expression in eukaryotes. So for any particular gene in a eukaryote, we're going to have a promoter, an enhancer, and then we might have a silencer too. An enhancer and silencer are exactly what they sound like. If there's an enhancer sequence, it can increase the rate at which a gene is transcribed, so how often it's transcribed. If we have a silencer, it can kind of deaden the effect of that gene. It can slow it down. The genes in eukaryotes are not typically all grouped together. There are some exceptions, but generally we don't have what we see in prokaryotes, where all the genes needed for one particular pathway are all grouped together. Instead, the genes are placed individually, usually with non-coding DNA sequences between them, and each gene has its own promoter and its own coding sequence. That's the part that's going to contain the information that's needed by the ribosome to make the protein. So what you're seeing here, we've got a promoter, and that's where RNA polymerase is going to bind. And then to the right of that, we have the coding region. That's going to be turned into an RNA transcript, which is a fancy name for a messenger RNA, and that's going to go out to the ribosome. Now there's all this other stuff going on here, and you can see the DNA is bent back in a loop. And basically, we have all these other proteins, these activators, these co-activators, etc., repressors. These are things that bend the DNA and give RNA polymerase a spot to bind. If all of these proteins are not present, then the gene will not be read. RNA polymerase will not know where to bind. It won't bind to the DNA, and it won't transcribe it. So kind of upstream to the left of our coding region here, we have these enhancers, and we have proteins that bind to that, and they bind to these co-activators, and then these bind to other proteins known as transcription factors, and all of those proteins need to be present. They need to find their place so that RNA polymerase can bind. Different cells will have different transcription factors, and a heart cell will have the transcription factors it needs to turn on certain genes that it needs. A liver cell will not have those same transcription factors, so that the liver cell will not turn on genes that are only needed in the heart cell. And then to top that off, if there's repressor proteins floating around, they can bind to the DNA and make this whole thing fall apart so that transcription doesn't occur. I know this all seems very complicated because, well, it is very complicated, but don't worry, we're not going to get into this in a ton of detail. I'm just going to point out some of the most important aspects of this. And we'll start with the structure of a eukaryotic gene. So we have the gene itself, that's the coding sequence that contains the information. Just before that we have a promoter, and that's the region where RNA polymerase will bind. Upstream of this, so to the left, ahead of the gene, we have this enhancer region, and activator proteins can bind to that. If those activator proteins are present in the cell, they bind to that enhancer region, and this causes the whole piece of DNA upstream of the gene to bend over. And then there's proteins that come along and they help maintain that bend. There's mediator proteins that bind to those activators, and then there's these general transcription factors that bind to those proteins. A transcription factor is just a protein that brings about transcription on another gene. So a transcription factor is a protein. It's going to bind to that site once this loop is made and everything's in place, and then RNA polymerase can bind to those transcription factors. But all this stuff has to be in the cell in order for this to work. When a cell divides, of course, the cytoplasm divides as well. So the transcription factors and all these different proteins are going to be divided between the two daughter cells as well. And that's how cells maintain their identity. That's how heart cells give rise to heart cells. And a heart cell is not going to give rise to a liver cell. So all this stuff has to be in place. We need to have all these proteins there. Everything needs to line up correctly. And if it does, then we have a place for RNA polymerase. And RNA polymerase will bind, and then we get our transcription occurring, and we get a messenger RNA being produced. Again, these transcription factors, all of these proteins, look for specific sequences, and different transcription factors look for different sequences. So we have to have the correct transcription factors in a particular cell that are needed to turn on the genes that that cell needs to use. Transcription factors are coded for by genes themselves. So there are genes on your chromosomes that code for transcription factors, and then they go off and they activate other genes, or they might turn off some genes. One transcription factor might do both. It might turn on one gene and turn off another gene. So it might act as an enhancer for one gene and a silencer for another, for instance. Genes that affect lots of other genes are known as master genes. So we can have a small mutation in a master gene that codes for a transcription factor, and that can affect hundreds of other genes within the cell or within the organism. What you're seeing here, without getting into too much detail, there are master genes known as Hox genes that are turned on only during early development. And they basically tell the different regions of your body what their identity is going to be. So you have certain Hox genes that are turned on just in your thorax, in your chest. And those genes turn on the genes that are needed to make heart cells, and turn on the genes that are needed to make the structures of the lung, etc. So we have these genes that are turned on quite early in development that tell the embryo this is going to become the head, this is going to become the neck, etc. And they go about turning on or turning off a bunch of other genes so that the cells get this identity. And we'll come back to that in just a bit. I find the effects of these genes especially interesting in insects. What you're seeing here at the top is what we think the ancestor of all known insects today came from. So we started out with an organism that had lots of legs and segments that looked all the same. Kind of like a centipede today. But it's thought that something like that gave rise to insects and also crustaceans and centipedes and millipedes and so on. Within an insect, we have three well-defined regions. We have the head, which is shown in green here. We have the thorax, which is where the legs and the wings attach, shown in yellow. And then we have the abdomen, which is shown in orange and red. And what seems to have happened is that certain master genes got turned on in each of these regions. And within the head region, these particular genes tell the legs up the front here to develop into mouth parts. If you've ever had a really close look at the mouth of a grasshopper, they've got all these weird appendages there, and they're actually modified legs. If we look at the thorax, we've kept the legs there, although they've become modified as well. And if we look at the abdomen, there's genes that were turned on during evolution that turned off all the genes needed to make legs in that region. Interestingly, they're not turned off in the larva sometimes, so caterpillars do have these kind of stumpy little fleshy legs in the back, but for adult insects, we don't see legs within the abdomen. Interesting things can go wrong if we get mutations in these master genes. So they're going to affect development in some really profound ways. So at the top there, you have the face of a fly, and this is not a normal fly on the right here. On the left, we have a normal fly. We have little antennae here. Now, antennae are actually modified legs. And you can see the fly on the right has legs on its face. Like that is a fully formed leg on its face. So there's a gene, don't worry about the name of this, but it's called antennapedia, and it's normally turned on in the head, and it basically tells that appendage to develop as an antenna. So it turns on all the genes needed for an antenna and turns off the genes that are needed for a normal leg. And that appendage develops as an antenna. If that gene is not working, so if that gene is completely broken, then the default pathway for that appendage is a leg, and it develops as a leg. So you can get flies with legs on their faces, and it's just due to this one gene not working. The picture on the bottom there shows a fly with two sets of wings. That is unusual. We've got a thorax here, and we've got a second thorax here. So we should only have one thorax and one pair of wings. Now what's happened here, we've got another gene, a master gene, that was turned on in the wrong spot. And it told one of the segments to develop into the thorax instead of becoming part of the abdomen. Again, we have the same master genes in us. You can see that with these Hox genes, we have 4 of them that are turned on in the head, and they're responsible for initiating the development of the brain and other structures of the head. We have some that are active within the thorax, and they bring about the development of the organs we see in the chest. We have some that are expressed in the limbs. We have some that are expressed in the neck, and they bring about the identity of those regions. Again, the take-home message here, it's a lot more detail here than I expect you to know. The take-home message is that genes like this code for a protein that's a transcription factor that turns on a lot of other genes. And then those genes might be transcription factors too, and they turn on a lot of other genes. So one gene can have a huge effect on development. Moving on to the next way that eukaryotic cells can control gene expression, and that's something known as alternate RNA splicing. Something we won't get into in a lot of detail, but quite fascinating nonetheless. I think I've mentioned before that it's thought that humans have about 24,000 genes. That's actually not enough to code for all the different proteins we have. What happens is within our genes, we have these areas here. They're known as introns or interruptions. These blank areas here don't actually code for anything. The other areas here that are colored in, they're known as exons, and the exons are used to make protein. So the boxes there that I'm showing with the arrows, those are coding regions within a gene, and then they're interrupted by these non-coding regions. What happens is one big messenger RNA is made, and then those non-coding regions are cut out, and the other pieces are spliced together, and that gives us our messenger RNA. But what might happen in one cell is that maybe all of this is going to be cut out too, and then perhaps we can get a messenger RNA that just has these 3 codons here. In a different type of cell, maybe we're just going to use, I don't know, let's use this one and this one and this one down here. So basically, these coding regions serve as like Lego blocks that can be spliced together to give us a different messenger RNA that will code for a protein that's a little bit different. So in the past, I've said that one gene codes for one protein. It's actually more complicated than that in eukaryotes. Not in prokaryotes. Prokaryotes don't do this. But in eukaryotes, it is possible to have one gene that can code for a number of similar but slightly different proteins. We also have control at the level of translation. So we can stop a ribosome from binding to a certain messenger RNA. On the ribosome, there's actually a sequence of RNA that the messenger RNA binds to. It has to be the right sequence for that particular messenger RNA to bind. So here's just another opportunity for the cell to shut down a certain gene. The gene is still transcribed, but that particular messenger RNA is not going to be read. Finally, we have post-translational modifications. So after the polypeptide is manufactured by the ribosome, there's still things that can happen to it. It's not done yet. So we can add carbohydrates. Most enzymes and channels and so on have to have some carbohydrates added before they're active. We have cleavage of amino acids. Cleavage is just a fancy term for cutting. So we can cut off some amino acids and change the protein after it's actually manufactured. We can have the binding of cofactors. So vitamins, for instance, a number of the vitamins that you take if you're taking vitamin pills are needed to activate certain enzymes. And sometimes ions like magnesium, for instance, are needed to activate certain enzymes. If that ion's not present, the enzyme's not going to do anything. And then also, of course, the cell can decide where that protein goes. We have lots of compartments within the cell. They have different pHs. That's going to affect the shape of the protein and whether or not it functions. So just looking at this very quickly, remember that we have a lot of very large complexes of proteins in cells. One example would be ATP synthase, which manufactures ATP in the mitochondria. Very complex collection of lots and lots of proteins. So we can take proteins, bring them together and build something. We can take some of the same proteins, mix them with other proteins and build something different. Proteins often need to be glycosylated before they become active. Glycosylation is just a fancy term for adding sugars. And the example we've used before are the antigens, the proteins that stick out on the surface of red blood cells that identify your blood group. This glycosylation begins in the rough ER and is continued within the Golgi. And the Golgi ultimately decides where those proteins are going to go afterwards. So a lot of proteins, before they're fully active, they need to have sugars added. Sometimes we need to have more than one polypeptide come together before the protein is active. And in some cases, we actually have to have a chunk of the protein removed before it becomes active. What you're seeing here in blue is trypsin, and this is an enzyme that breaks down proteins, and it's manufactured within the pancreas. Now, if the pancreas were to manufacture this trypsin and fill up its cells with trypsin, the trypsin would attack those cells. It would break down the cells that make up the pancreas. That wouldn't be very good. So it needs to be secreted in an inactive form. It's got this short little chain of amino acids that acts kind of like a safety switch. So when that's removed, then trypsin can take its final form and have a fully formed active site of the correct shape, and it can do what it needs to do. So inside the pancreas, it has this inactive form. It's released through the pancreatic duct into the duodenum. That's the first part of the small intestine. And there's enzymes there that cut off that little safety thing. So they remove that small chain of amino acids, and now trypsin can become active where it should be active, where it's going to attack the protein that you took in with your food and not the protein that makes up the cells that manufactured it. Many proteins need to have cofactors before they're fully functional. What you're seeing here is hemoglobin. It consists of 4 polypeptides. Each of those polypeptides doesn't do a whole lot on its own, so we need to have those 4 polypeptides come together. That would be the quaternary structure of the protein. But we also need to have iron. So iron is going to be added, and iron is the site where oxygen will bind. So hemoglobin is used by your blood to transport oxygen around your body. But it can't do its job until all those 4 pieces are in place and until these 4 iron atoms are added as part of a heme group. Most proteins need to be in a particular place before they can do their job. So an example of that would be insulin. Insulin is manufactured by the pancreas. Pancreas does a lot of wonderful things. Insulin is not dumped into the digestive tract, though. It's dumped into the blood. Insulin doesn't do anything in the cells that produce it, but after it leaves those cells, it seeks out other cells, it binds to receptors, it acts as a hormone, and it brings about changes in those cells. On the protein that's spat out by a ribosome, there may or may not be a short sequence of amino acids known as a signal peptide. If there is, then there's this protein that grabs onto it and brings it over to the rough ER. And what's going to happen is the ribosome is going to dock with this channel, and the protein will end up getting dumped inside the ER. Now again, that's only going to happen if there's a signal peptide. If there's no signal peptide, then the protein gets dumped into the cytosol. But if there is a signal peptide, then that protein gets dumped into the inside of the ER. It's probably going to go to the Golgi after that, and then it may end up embedded in the membrane, or it may end up being secreted. But this signal peptide will determine whether or not that's a possibility. Eukaryotes do indeed have a lot of different ways of regulating gene expression, of regulating whether or not a certain gene is turned on in a certain cell, and whether or not that gene, even if it is turned on, is going to have an effect. For prokaryotes, things are definitely quicker and easier, and they can do stuff faster in some cases because of that. But having this increased level of complexity does have some benefits. In multicellular organisms, we have eukaryotic cells that have to take on very specific roles. So just in your body, you have over 300 different types of specialized tissues, collections of cells that do specialized things like contract or digest or whatever. We can turn on some genes in some cells and turn on different genes in different cells so that those cells can have different identities and do different things, even though they have the same library of information. And this is what has allowed multicellularity to evolve. We start out as a single diploid cell, the zygote, or fertilized egg on this diagram. Remember, that came from the fusion of a sperm and an egg cell. That cell is very unspecialized. It divides into 2 cells, that divides into 4, into 8, etc. And even at that 8-cell stage, those cells are still unspecialized. But as the embryo divides further, certain sets of genes are turned off permanently, certain sets of genes are turned on. We have transcription factors that are involved in that are passed down with the cytoplasm to the daughter cells, and they maintain that identity. So when we end up with a fully formed human, we have all these very specialized cells that have this identity built in. All those cells have the same information. Think of it, again, like a library, but certain shelves have been locked. So certain books on the chromosome have been locked away, and only certain genes are accessible. That's typically the case. Now, cancer cells, we'll talk a little more about cancer in the future. They can turn on some of the genes that they shouldn't be turning on. But typically, these specialized cells have some genes that are turned off permanently and some genes that are very active. And different cell types will have different sets of genes that they use. And our concepts. So enzymatic pathways are controlled at the protein level by non-competitive interactions and negative feedback inhibition, as we've talked about. That's not as efficient, though, as controlling the pathways at the genetic level. And that's something that prokaryotes do very, very well. So prokaryotes have these operons where all the related genes that are needed for a particular pathway are all grouped together, and they're all under the control of one promoter. Eukaryotic genes are not clustered into operons. Again, things are messy. The genes are kind of all over the place typically. There's some exceptions. The Hox genes are all right together, which is kind of interesting, but for the most part, the genes are all over the place. The genes are transcribed and translated individually, and we do have some genes that code for proteins that regulate other genes. And in the case of master genes, we might have one gene that affects hundreds of other genes. In order for something to be secreted, a protein to become secreted, remember it has to move into the endoplasmic reticulum, and that is going to only happen if there's a signal peptide. That's one way that we can regulate where a protein ends up and what it does, but we can regulate gene expression in all those steps that occur within the cell. And the most important regulatory step is translation itself, whether or not we have the correct transcription factors present. And finally, our terminology.